The Cell by Ashton Kelly, Ella Gray, and Justin Jones. The cell membrane is a thin, semi-permeable membrane that surrounds the cytoplasm of a cell, enclosing its contents. Its function is to protect the integrity of the interior of the cell by allowing certain substances into the cell, while keeping other substances out. It also serves as a base of attachment for the cytoskeleton in some organisms and the cell wall in others. Thus, the cell membrane also serves to help support the cell and help maintain its shape. The fluid mosaic model is a model that describes the structure of the cell membranes. In this model, a flexible layer made of lipid molecules is intersected with large protein molecules that act as channels through which other molecules can enter and leave the cell. Continuing on the fluid mosaic model, we have membrane proteins, which provide functionality to the membrane. As their name suggests, membrane proteins are anchored in membranes. However, each of them has a specific function that often requires them to extend beyond the membrane. Some membrane proteins sit on the surface, while others are anchored in the membrane and have pieces on one or both sides of it. Proteins with extracellular domains are usually involved with cell-to-cell communications or interactions. Proteins that reside mainly within a membrane usually form channels or pores that allow molecules across the membrane. In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein bind only to a specific molecule, such as a sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules on their concentration gradient, and this movement is carried out without any input of energy, which makes it different from our next topic, which is active transport. Active transport is a kind of transport where ions or molecules move against a concentration gradient. This means that there's movement in the opposite direction of diffusion. This movement against the concentration gradient will require energy, which makes it very different than passive transport. Hence, this process will use the assistance of a type of protein called a carrier protein, which uses ATP for activation. The next section is the nucleus. In biology, the major functions of a nucleus are to maintain the integrity of DNA and to control other cellular activities such as metabolism, growth, and reproduction, by regulating gene expression. It has three main components: the nucleolus, the chromatin, and the nuclear envelope. The nuclear envelope has nuclear pores to control the movement of molecules between the nucleoplasm and the cytoplasm. Each cell contains this control center with instructions on how to run day-to-day operations and specialized functions. These instructions are your DNA, which is stored in its very own cellular glove compartment, the nucleus. The nucleus is a membrane-bound structure that contains the genetic material of a cell. Not all cells have nuclei, but many cells, such as those in plants, fungi, animals, and protists, contain these structures. Next up is the endomembrane system. The endomembrane system is a group of membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins. It includes the nuclear envelope, lysosomes, and vesicles, and the endoplasmic reticulum, and lastly, the Golgi apparatus. Although technically not within the cell, the plasma membrane is included in the endomembrane system because of the way it reacts with other organelles within the membrane. In order to further understand the endomembrane system, we need to understand how secretory and membrane proteins are made using the endomembrane system. The membrane and secretory proteins are synthesized in the rough endoplasmic reticulum. The rough ER also sometimes modifies proteins. In the illustration to the right, an integral membrane protein in the ER is modified by the attachment of a carbohydrate. Vesicles with the integral protein bud from the ER and fuse with the cis face of the Golgi apparatus. As the protein passes along the Golgi cisternae, it is further modified by the addition of more carbohydrates. After its synthesis is complete, it exits as integral membrane protein of the vesicle that bud from the Golgi's transphase. And when the vesicle fuses with the cell membrane, the protein becomes an integral portion of that cell membrane. Next up is the lysosome. Now basically what all those words to the left mean are that lysosomes are cellular organelles that contain acid hydrolase enzymes that break down waste materials and cellular debris. They've been called the garbage man of the cell because they basically digest old organelles, food particles, and other engulfed viruses and bacteria through phagocytosis. Now moving on, the next portion is a peroxisome, which is essentially the exact same as a lysosome, but they're both special in their own ways. So the difference between a peroxisome and a lysosome is that with peroxisomes, they break down larger harmful molecules into smaller useful molecules. A peroxisome, or an example of a peroxisome doing its job, is that in the cell, it will break down hydrogen peroxide into oxygen and water to prevent it from harming the cell. Next up is the centrosome. The centrosome is located in the cytoplasm, and its function primarily is to help set up the location of the nucleus as well as other organelles in the body. The importance of centrosomes is that besides setting up the location of the nucleus and other fun organelles, they aid in mitosis via the creation of the mitotic spindle. The mitotic spindle allows it to separate the chromosomes, which is very important in our creation. So a product of the centrosome is actually the cytoskeleton. The cytoskeleton holds all the organelles inside of a cell in place. The importance of the cytoskeleton is that it provides shape, movement, and anchorage for cells. Without the cytoskeleton, a cell can lose essential functions and damage the cell. And depending on which portion of the cytoskeleton you use, whether it be microfilaments, microtubules, or intermediate filaments, each has its own specific role in the cell, whether it be resisting compression, holding things in place, or allowing muscle movement. Moving on from the cytoskeleton, we're moving on to the mitochondria, which is the power pack of the cell. Basically, the mitochondria is known as the powerhouse of the cell because it plays a prime role in ATP formation and cellular respiration. Once the Krebs cycle is completed in the cytoplasm, it moves to the mitochondria where we can move on to our electron transport chain and make all that fun ATP, which is vital for providing our cells with a lot of energy. Well, that about sums up our tour of the cell. Thank you so much for watching and have a great day.
That was great.